Hi everyone and welcome to GCSE Revision with MFS. In this video, we look at the required practical for food tests. We focus on the method used to carry out this required practical and the key observations you would see. Please ensure you hit the subscribe button and the notification bell to ensure you never miss a video from GCSE Revision with MFS. Okay, so here is everything you need to know for the required practical. You need to be able to use qualitative reagents to test for the range of carbohydrates, lipids, and proteins. The tests we are going to focus on today includes the Benedict's test, iodine test, and the Biuret reagent test for proteins. Okay, so the first steps of the method include breaking down your food sample and dissolving it so you can see a visible color change to show a positive or negative result. So you would first of all use a pestle and mortar to grind the food products. So you can break the food products down into smaller, more soluble pieces. Then you would transfer the products into a beaker and add some water as well as stir the solution. This will allow the food products to dissolve into the water. And any insoluble food molecules you don't want in there, so you would use a filter paper, filter funnel to filter out the insoluble pieces of the food sample, and then in a conical flask you will have the solution. Here's an image of what a pestle and mortar look like. You would use this to grind your food products into smaller soluble pieces. Now, once you have your solution in your conical flask, you can see a more visible color change. Here are the results you would see, and it's really important you learn the test for each of these food groups and the results you would get for a positive test. So for starch, you would add an iodine solution to your food sample and a positive result would be shown by the color changing blue or black. When you're testing for sugar, you would add in Benedict's solution and heat the solution with the food sample and Benedict's solution in a water bath. And for a positive result, you would see a change in color to green, yellow, or brick red. And the color depends on how much sugar is present in the solution. For proteins, you would add in Biuret solution and a positive result would show a purple color. And for lipids, you would add in ethanol, and for a positive result, you would see a white cloud emulsion forming. Now, the key word over here is emulsion. If you don't try emulsion in an exam, you wouldn't get the mark.
Okay, so hopefully now you should be able to explain how to carry out the required practical for food tests. Thank you for watching this video. Please hit the like and subscribe button to ensure you never miss another video from our channel, and I hope to see you in the next video.